Hi, everyone. Thank you for joining the 8th webinar in our series on the use of NAMs in risk assessment. This series is being co-organized by the EPA, the Physicians Committee for Responsible Medicine, and the PETA Science Consortium International. My name is Amy Klippinger. I direct the PETA Science Consortium International, and I'll be co-moderating today's webinar with Todd Stedford, who is a Senior Science Advisor at the EPA Office of Pollution Prevention and Toxics, and Christy Sullivan, who is the Vice President of Research Policy at the Physicians Committee for Responsible Medicine. We have two speakers today who will be presenting on the use of computational and AOP-based approaches to inform carcinogenicity assessments. First, we'll hear from Martin Phillips of the EPA Office of Pollution Prevention and Toxics, and then we'll hear from Susanne Stalford of LASA. Before introducing our first speaker, I'd like to point out that the slides from today's webinar can be found online at the link on the screen, and a recording will be posted there as well shortly after the webinar. Slides and a recording of the first 7 webinars in this series are also available at the same link. There will be time for questions for both speakers at the end of the second presentation. Everyone is on mute, but you can type any questions in the questions section within your GoToWebinar toolbar, which should show up on the right-hand side of your screen. If you send in a question, please note if your question is for a specific speaker or for both. I'll now turn it over to Dr. Todd Stedford for his introductory comments. Todd?
Thanks, Amy. Let me start by saying I'd like to thank PETA and PCRM for their help and logistical support. I'd also like to thank the presenters for their time and the attendees for their interest in today's presentations. As all of you are aware, probably, EPA is committed to its work towards implementing NAMs. OncoLogic is part of that work and was recently included in EPA's updated NAMs list since our last PEP webinar. Speaking first today on OncoLogic version 9 will be Dr. Martin Phillips. Martin is a chemist in EPA's Office of Pollution Prevention and Toxics in the Data Gathering and Analysis Division. Martin joined the agency in 2019 after working at the Minnesota Department of Health and at Cytovation, with his past experience focusing on computational toxicology and the development of new approach methodologies. Martin's work at EPA encompasses data science, new chemicals evaluation, participation in national and international work groups, and systematic review activities. And I'll now turn it over to Dr. Phillips.
Thank you, Dr. Stedford. I appreciate the introduction. I would like to my presentation today is about OncoLogic version 9 and its use at EPA in the New Chemicals Program under TSCA. OncoLogic is a mechanism-based expert system for predicting carcinogenic potential. It was developed by domain experts at EPA and elsewhere in collaboration with an expert system developer, which is a type of computational tool. OncoLogic encodes knowledge from structure-activity relationships on over 10,000 chemicals. There's a class-specific approach to optimizing the predictive capability of the OncoLogic system. So each chemical class is handled separately with its own rules. We consider relevant factors, including biological input when possible, such as the results of in vivo and in vitro genotoxicity testing. Predictions come with a scientific rationale and a semi-quantitative ranking. How does it work? OncoLogic mimics the thinking and reasoning of a human expert using knowledge-based rules for classes to predict cancer concern. Each chemical class has a baseline level of concern ranging from low to high. Then it looks at substituents on the chemical that may affect the carcinogenicity. The concern level changes accordingly. We call these rule makers or rule busters. There are many benefits of using OncoLogic It expedites the decision-making process, allows for sharing of knowledge amongst assessors, reduces errors and inconsistencies amongst the assessment team, helps formalize knowledge rules for cancer hazard identification, and allows non-experts to reach scientifically supportable conclusions. There are 6 different concern levels that OncoLogic can assign, ranging from low to high. A low concern level is a chemical that is unlikely to be carcinogenic. Marginal is likely to have equivocal carcinogenic activity if run in a 2-year cancer bioassay. Low to moderate is likely to be weakly carcinogenic. Moderate is moderately active. Moderate to high, highly likely to be a moderately active carcinogen, and high is highly likely to be a potent carcinogen. EPA now runs all new chemicals through our computational systems, including the QSAR toolbox and OncoLogic when there's cancer concerns. Before using OncoLogic in the new chemicals workflow, we had class-based structural alerts like aromatic amines to predict potential cancer hazards. This is a refinement on that system. So a new chemical substance comes into the agency as part of a PMN submission or low volume exemption submission. The submission goes to discipline-specific reviews. So there are 6 different disciplines: ecotoxicology, human health, fate, chemistry, engineering, exposure. And when it gets to the human health discipline, we look at structural alerts in order to, as part of our hazard identification process. An example structural alert might be an aromatic amine. We can then enter the structure into OncoLogic version 9, usually using the SMILES code. If there's a low concern, then we can reduce the concern for cancer. If it's a result other than low, then we can start doing an analog search or slope factor selection for a quantitative risk assessment or a weight of evidence evaluation. So again, low is one of the 6 different outputs of the OncoLogic system. So previous to this, we would identify hazards for broad chemical classes, and this is a refinement of that by using, again, information on over 10,000 compounds and structure activity relationships, expert judgment, to try and refine those broad-based structural alerts. So at this point, I'm going to do a demonstration of the OncoLogic system. So the OncoLogic system can be downloaded from our website. We actually have 2 versions of OncoLogic that are available. OncoLogic version 9, which is the current version that I'm going to be demoing today, and OncoLogic version 8. OncoLogic version 8 was much more difficult to use. It didn't have a intuitive approach for entering chemical structures, and it was very difficult for people without a knowledge of chemistry to apply version 8, because you had to know a lot about interpreting the chemical structure and assigning the initial chemical class in order to even get started with an SAR. So the version 8 looks like this. There are 4 different modules in version 8. Fibers, polymers, metals, and organics. So one of the reasons why we still are supporting version 8 for a while is because the fibers, polymers, and metals modules have not yet been ported over into version 9. But the organics module is the largest module, and it's the one that requires the most input. So I'm just going to give an example. So you have to select the appropriate class of compounds from this long list. So someone without a lot of chemistry knowledge may not be able to tell if their compound is a coumarin, a furocoumarin, or an epoxide. So there are some questions, including what is the molecular weight of the substance that you have to answer? What are the routes of interest? A skeleton, and then you get into the chemical entry where this is, you can tell this is not a standard interface for entering chemical structures. By comparison, OncoLogic version 9, you can enter chemicals by CAS number, by chemical name, or by structure. In order to use the CAS number and chemical name features, you will need to have the OECD QSAR Toolbox version 4.3 co-installed on the same system, because it uses the OECD QSAR Toolbox database in order to do the CAS number and chemical name lookups. You can always enter something by structure or SMILES. So as an example, I'm going to look up CAS number 62759. So you click search. This is our result. You click evaluate. The chemical class is assigned automatically by OncoLogic. And then you can click evaluate, and it will give you the overall concern level that we discussed earlier. In this case, it's a level of high. It also develops this justification report that shows the chemical structure as entered and a justification, including factors that are known to increase or decrease the carcinogenic potential and the overall concern level. So going back to the input, we can also search by chemical name. I'm going to search for 1,2-dibromopropane. So you can see it comes up with the correct chemical structure, or you can draw a chemical structure. When you click the edit button, you'll get this edit structure dialog box where you can either draw it using more intuitive chemical drawing tools, or you can copy and paste a SMILE string into your dialog box. So I'm going to draw the structure. Okay, it assigns this to the aldehydes chemical class. When we click evaluate, we can see what the level of concern is for this compound. So here it shows marginal to low moderate. In this case, the aldehydes chemical class develops different levels of concern for different routes of exposure. It runs all of these automatically. You don't need to enter them separately. So it will develop a dermal route of exposure. So this is marginal, inhalation, low to moderate, injection, low to moderate, and oral, marginal. So you can see that there's actually different levels of concern for different routes of exposure. And then if you click on the gavel icon, then it gives you the overall level of concern, which is marginal to low moderate. For certain chemical classes, you will actually have additional prompts. So I'm going to draw one more structure here. So this is in the trialkyl phosphates category. So it asks, I'm going to try and magnify this. It asks, select the results in vivo genotoxicity testing. So if you have data that shows that it's negative in vivo, you can click that. Select the results of in vitro genotoxicity testing. Let's say it's positive in vitro. Is rapid hydrolysis or detoxification expected? Yes, no, or unknown. Let's just say unknown. Then it's going to give you the overall concern. So in this case, it's low to moderate. So it also incorporates the answers that you put in here as part of the justification. So in this case, the justification report says, even though the compound is genotoxic in vitro, in vivo genotoxicity testing results were negative. Furthermore, the compound may undergo rapid hydrolysis or detoxification. The level of concern for compounds with these functional properties are expected to range from marginal to low moderate. So this has actual input based off of the results of testing that you can add, and you can specify yes, no, or unknown for many of these questions. The questions that you get when you use the program depend on the chemical class. So the organophosphorus class is an example of a chemical class where you get all of these questions about hydrolysis, genotoxicity testing results, etc. As we saw earlier with the aldehydes, when I clicked evaluate, there weren't any questions that popped up. So it's class specific. Okay, let's get into a little bit more of the detail of how OncoLogic works, or the different classes. So I'm gonna click on my positive button again. So up in the upper left-hand corner next to chemical class, you can click this little eyeball button. This brings up the profiling scheme browser, which is not responding. Okay, it did respond, yay. Okay, so you can see all of the different chemical classes that are built into OncoLogic. So under acylating agents, there's acyl and benzoyl halides, anhydrides, carbamyl halides, and phosgene type compounds. Under aromatic amines, there's a large number. So you can see all of the different chemical classes. And then you have a decision logic in here. So let's go to one where it's a little bit more interesting. So in this case, we have two different rules that are combined by an OR operator. I'm gonna have to zoom out so you can see how these work. So if you click on rule 1, you can see down here at the bottom that there's a 5-membered ring with a carbon here. If you click on rule 2, then there's a 7-membered ring. So these are known as smart strings. So these are describing the structural alert in order to figure out which chemical class the chemical belongs to. So all of this is transparent because you can see exactly how the software is assigning the chemical class. Now, if you click on, I'm going to go back into the magnified view. We're going to go over to this upper left-hand corner and click on the little eyeball next to the actual chemical class that it assigns. So in this case, it's a trialkyl thiophosphate. So this now shows the decision logic for this chemical class. So you have the chemical class trialkyl thiophosphates, baseline level concern, concern neutral substituents, concern reducing substituents, concern raising substituents, and then the final concern level. We can see a list of, if you click on any of these boxes, you can see what the concern reducing and concern raising substituents are. So you can see exactly how the decision logic is built. And then in the lower left-hand corner down here, you can actually see your chemical structure and which rules were applied. So it shows, in this case, the group is attached to an oxygen or sulfur heteroatom. So that was one of the ones that was applied. And then you can see there's this X, this red X that shows that this one was not applied because there aren't any vinyl groups. So you can go in and see exactly which rules were applied, which ones raised the concern, which ones lowered the concern, and what the overall pathway for the decision is. So in this way, we're hoping to increase transparency for the predictions. One more thing I wanted to show. So if you have a chemical that is part of more than one class, it will generate predictions for all of those classes. So in this case, we have both a nitrosamine and an aldehyde. So you can see now it's generating 2 levels of concern, and you can evaluate each one independently. And then you go with the level of concern that is highest. So I'm going to zoom in again. So for the nitrosamine, this is giving a moderate level of concern. For the aldehyde, it's giving a marginal to moderate, and the aldehyde depends on the route. So it's moderate via inhalation, but marginal via dermal. And another feature is it shows your chemical structure as entered in the corner here. And the green highlighted groups are groups that couldn't be processed under that chemical class. So under the nitrosamine class, all of the atoms are black. So it was able to evaluate the whole molecule. And each of these, basically what that means is all of the substituents were substituents that the decision logic was built to handle. When you click over to aldehydes, you can see the nitrosamine section is green highlighted, which means that it wasn't able to determine how that group affected the concern level for this class. So that's one of the reasons why we evaluate under multiple classes at the same time, so that we can make sure that we get the best prediction. And it provides a little, in the case of the aldehydes where we have this green group highlighted, you can see this exclamation point here where it says the effect of any highlighted substituent or Z group is uncertain. So it will let you know that that is a concern. Okay, one last thing. For those of you who might not have the OECD QSAR toolbox already downloaded on your computer, if you need to search something by CAS number and enter, you can go to PubChem, type in the CAS number, and then scroll down to the SMILES code, which is in section 2.1.4 of PubChem. Copy that. And paste it into our draw structured dialog box, and it'll just draw it. So that's an easy way to draw large molecules, or if you don't have the QSAR toolbox co-installed on the same system, then you can do it as a SMILES entry. Okay, thank you so much. That's the end of my presentation and demo.
Okay, thank you, Martin. That was an interesting demo. You certainly made it look easy. It does seem like version 9 is quite user-friendly and intuitive. So thanks very much for going through that. And then as a reminder, we will have time for questions, but we're going to hold them all to the end. Please start typing them in now, though, as you have them using the GoToWebinar questions panel, and we'll ask them after our next presentation. And as you send in your questions, please note if your question is for a specific speaker or for either. Speaking next will be Dr. Susanne Stalford. Susanne received her master's and PhD in chemistry from the University of Leeds in the UK. She has worked as a scientist at LASA for 13 years, working on a multitude of projects and products, including working for the past several years as part of a team developing adverse outcome pathways. The title of her talk today is Development of an Adverse Outcome Pathway Network to Support an Integrated Approach to Testing and Assessment of Carcinogenic Risk. Susanne?
Hi there, thank you for the introduction. So today I'm going to talk you through a bit of work we are doing at LASA Limited around developing adverse outcome pathway networks for carcinogenicity and looking at how these could be used and support an assessment for carcinogenic risk. So hang on, there we go. So I'm going to talk a bit about the work we're doing. So I'm going to introduce by looking at what the future of safety assessment looks like in relation to CARC testing. And then I'll talk about the approach we are developing to aid CARC assessment using alternatives. So in our work, we've been looking at adverse outcome pathways as the basis of our concepts. And then we've looked to our current in-house knowledge and using that in a different way to form our AOP network. And then look at how we can link evidence to our framework and discuss how this can be combined to give a potential prediction as such, or an assessment to help support your further assessments, and then talk you through an example and potential use case. So to start, I thought I'd give you a view of where safety assessment is going. So right now, many regulations require the use of in vivo studies to give the ultimate say as to whether a substance being brought to market will have an adverse effect. However, there are already moves to reduce animal testing in recent years, and an example of that being the ban in Europe of animal testing for new cosmetics. So this regulation works because there are viable, well-developed in vitro alternatives available that are proven to be reliable for extrapolation to that in vivo scenario. But this isn't the case in many other areas. So the hope is that as time progresses, in vitro and in silico alternatives are developed in such a way that they are dependable in predicting the outcomes of animal testing. So in vivo studies will only be required when outcomes in alternatives are unclear or just used in a targeted or confirmatory manner. So eventually, we might get to a point where animal testing is not required for any substance at all. And with the development of highly predictive and more sophisticated computer technologies, the need for in vitro testing would potentially also decrease. So the prediction of human CARC has always been a principal area of concern during substance safety assessments in a wide range of industries. And this includes agrochemicals, industrial chemicals, pharmaceuticals, and veterinary medicines. And this usually requires the use of the 2-year rodent bioassay, which is done in accordance with OECD Test Guideline 451. And this is used to ultimately determine human risk. However, there are many reasons why alternatives should be looked at. So firstly, the time it takes to conduct a study. So this is a lifetime study, so it's 2 years for rodents and could be longer for other species. There's the cost to conduct a study. There's the ethical consideration. So the number of animals at the moment recommended for testing is over 50 of each sex at each dose and control group for each species tested. And because at least two species is usually tested, that means hundreds of animals are needed. There's also the concern about human relevance. So in a rodent model, not all modes of action are relevant for humans and vice versa. So there's potential for both false positives and negatives when extrapolating from rodent to human risk. So we think that replacing a 2-year rodent assay is of paramount importance. So just to touch on alternatives, there are already some alternatives which can be used to help predict human CARC risk. So as I work for LASA, here is an example where we show the in silico models which can be used to satisfy the ICHM7 regulations, which allows the use of 2 in silico mutagenicity prediction models, one which is expert derived and one which is statistically derived, to classify an impurity concern in an active pharmaceutical ingredient. So with this workflow above, we have our 2 systems, which is Derek and Sarah, but also there's various other resources in prediction models we have as well, which could help, for example, predict the degradation of compounds and what they could break down into, purge evaluation, so you can see how much impurity may be in your drug, and various databases as well. So this already kind of shows an example of how in silico models can help with assessment without having to test in animals. There are also other alternatives out there, which include regulatory approved in vitro assays, which can act as indicators of genotoxic carcinogenicity and are already used for risk assessment. But there are also other up-and-coming models, such as gene expression assays, use of zebrafish, use of organotypic models, or there's also the high throughput mechanistic assays. However, these never all on their own or when in isolation can give that full picture of what might go on in human risk. So they may provide indicators of what might happen, but you don't get to see the whole picture. So this moves us on to an IATA. So there's concerted effort in many industries to move towards an integrated approach to testing and assessment, as much more testing is done using alternatives, and the amount of evidence available for substances is larger. So as I said, a test in isolation may not give the full picture when it comes to carcinogenic risk in humans. So if you compile all that different data, this is the most likely way that traditional animal models will be replaced. However, with evidence coming from many different sources, how can an overall conclusion be reached? And this, we think, is a significant challenge, and there's several factors which need to be considered. So what does the assay or test you're using actually measure? So how closely is the assay or test you're conducting linked to the adverse outcome you're trying to predict? And how does the findings in the test or assay you're performing relate to findings in other assays and models? So, for example, we have this series of data here, ranging from in silico to in vivo studies. It's like, how would you relate an in silico finding which is positive to an in vitro finding which is negative? And how do you know which one is correct? Are they measuring the same thing? And which one is more potentially related to the adverse outcome you're trying to see? So we think adverse outcome pathways, or AOPs, can provide that framework to answer these questions. And in recently, the OECD have published a guidance document which outlines the fact that alternative methods to toxicity testing are emerging, and we need to be using this evidence coming from these assays in an IATA sort of fashion. So they propose adverse outcome pathways as being the perfect framework for catching and reasoning between both models and data in a meaningful way. So just before I move on to my next slide, my cat might decide to come and interrupt. So I'll apologise in advance if you get interrupted. So talking about AOPs, if you don't know what AOPs are, so these are frameworks which can be used to describe causally linked steps in a pathway, and these which can lead to an adverse outcome. So in our case, we want to build a network of pathways which describe the steps which lead to carcinogenicity. So each of the steps along a pathway are known as key events, which are measurable activities and thus can be linked to tangible data. And each of these fall into the level of biological organisation, which are more complex as you get towards the adverse outcome at the end of the pathway, as shown in the schematic. They also have links between each of these key events, and these are key event relationships, and the strength of these links can be described by biological plausibility, empirical evidence, and essentiality. So now to move on to a bit about the work, how we're doing. So where we start with AOP, building our AOPs, we can use Direct Nexus as a starting point. So for those who don't know, Direct Nexus is the expert-derived knowledge-based software, and this predicts for a number of toxicity endpoints, including carcinogenicity. This has been developed using both public and proprietary data, literature review, and expert assessments to produce a comprehensive knowledge base of information stored as alerts, which, as well as giving a prediction of the likelihood of toxicity, also gives a commentary with examples, potential mechanisms, and structure-activity relationships. For example, we can assess the carcinogenicity of benzophenone using Direct Nexus. So we input a query compound and process it. So this comes back with the endpoint it activates. The likelihood it's going to be likelihood it's going to activate that endpoint, the alert it activates, and the associated information. And this association information is the source of our AOP network. So, for example, within this commentary, you can pick out the adverse outcome, you can pick out a molecular initiating event, and you can pick out various key events which can be arranged into a pathway. So, for example, we can take the statements highlighted in Alert 707 and arrange them into an AOP for PXR binding and activation, which leads to carcinogenicity. We can also assign each of these events to a biological complexity level. So from the molecular initiating event statement, If we process our benzophenone again, we can pick out 2 events at the molecular level, which is the molecular initiating event of PXR binding, which leads to PXR activation. From there, we add a key event relationship to the cellular CYP upregulation key events, which derive from the statement that PXR is an inducer of specific CYP enzymes. And from this, we can keep linking key events together based on the commentary from direct nexus to give us a skeleton AOP. So with this in mind, and with this methodology in place, we embarked on a project to rearrange all the knowledge present in these alert comments into an AOP framework. Since some of the most developed endpoints in direct nexus relate to the adverse outcome of cancer, we decided that this might be the best place for us to start. So we investigate all the CARC-related endpoints outlined in the table from direct nexus 6.0.1. So each of the 310 alerts investigated was assigned to a molecular initiating event where possible and various key events, if these were mentioned in the alert description. This resulted in us linking the alerts to 85 different key events. So not all the alerts link directly to the adverse outcome of carcinogenicity. So for instance, alert associated mutagenicity stops at key event of inherited gene mutation. So at this stage in our research, we made the assumption that these key events could be linked to the adverse outcome directly, but we were aware that further investigation of these putative AOPs may prove otherwise. So with the skeleton network in place, each of the pathways was subjected to a detailed review. So this involved using literature references to provide biological plausibility, and in some cases, empirical evidence to support key event relationships. We also put in place controlled terminology, so this was to ensure consistency within our network, and the potential to facilitate interaction with external data sources. So this investigation has significantly increased the number of pathways and key events in our network. And so far, we have 37 MIEs which have been identified, which are associated to 37 AOPs. But the number of pathways is more, some molecular initiate events associated to the adverse outcome can go via a number of different potential routes. So our current work has provided a network with 19 AOPs associated with genotoxic AOPs and 18 with non-genotoxic CARC AOPs. So this network also has the potential to be expanded further using additional sources. So Derek is a decent source, but we can expand this further with additional sources. So just to take you back to the graphic I showed earlier, so this is a similar style as to how to show the before picture of our horizontal putative AOP from alert 707. So as you can see, we've expanded first stage AOP to include heterodimer formation and gene modulation before the AOP splits into 4 pathways, which illustrates the ubiquitous effects the change in gene modulation can have. So something we are working on is the ability to group similar key events into one node, so a high level view of the AOP can be seen. And if more detail is required, the key event has potential to be expanded to reveal more specific detail. So now we have that network, we can attach evidence to the relevant key events. So the types of evidence potentially include like models such as direct nexus, alerts which have both molecular initiating events and key events. So we've got assays and then measurements. So the assays we've included so far are more regulatory in vitro and in vivo assays, such as the Ames test, chromosomal aberration test, and micronucleus test, as well as some binding assays and rodent repeat dose studies. And also we have our in-house database Phytic, and we've tried to link evidence from there where we can. So the work we've done so far is actually captured in our latest software, which we like to call Captis. So this screenshot shows the AOP for the PXR pathway, which I've discussed in the previous slides, with appropriate assays associated to the key events. The key events in green are different shades to correspond to the level of biological organization. So, for example, we see cellular key events in lighter green compared to events that occur at the organ level. So the numbers on each of the assay association pathways indicate the number of measurements from that assay relate to that key event links to as well. So not all pathways terminate at the adverse outcome, but they can provide more supporting information for the AOP. So we have all this in place now, but how can we use it? So we think this has the potential to be applied to a number of scenarios cross-industry, with the application of this framework depending on the use case in question. So For example, if you're in a discovery phase, you might have very limited evidence, but you want to assess the activity again of your compounds against a target. And this is to help aid in the decision on prioritizing on dropping certain leads. In early development, you may have more evidence, and you want to gather more to increase confidence in the decision. And the AOP may help the user to understand which assays would be most predictive or informative to increase confidence in the prediction, and use this to make decisions about a chemical. And then in your late development and safety assessment phases, you probably have a lot of data available for a compound, and this may be enough to predict any adverse outcomes. So this can also help identify gaps in knowledge and show you where you may need to fill these so that you can make a persuasive argument for perhaps a regulatory submission. So we have all this evidence, and you have a question you want to answer, and you can place your evidence on this AOP to help rationalise results between different assays in order to reach a conclusion. So, for example, we visualise a portion of one of the gene toxicity AOPs, which leads to CARC, illustrating how much data you may have. So having this evidence associated in this manner means we can use the components in the AOP in order to help reason between the data and assess the weight of evidence in order to reach a conclusion. However, how do you bring that all together? So factors you may need to be considered when coming to an outcome could include the quantity of evidence you have available, the quality of data and models being used, what systems the evidence comes from. So for example, thinking about how you treat evidence from an in vivo system in relation to those from an in vitro or in silico model, would you have more trust in an in vivo result over other results? And then thinking about the position of the evidence on the AOP as well, and how you would consider results from evidence closer to the adverse outcome than those earlier in the pathways. So just to give you a brief example, we can illustrate how to make a hypothetical assessment of clofibrate using the AOP framework. So here I've identified 3 AOPs which clofibrate could go through to cause CARC. So you have your genotoxic interactions, you have your H2HR binding, but then you also have your PPAR binding, and all these lead to the CARC adverse outcome. So as a preliminary, evidence has been gathered, and we've associated this evidence with the molecular initiating events and key events on the pathways. And you can see where these have been associated by the boxes with purple borders. As you can see, it is organised so it should be easy to make a logical, robust and transparent decision. So to decide on what the potential outcome could be, I've looked at this by analysing the evidence on each AOP and bringing the results together to give an overall assessment. So I'm looking at this perhaps from more safety assessment kind of area. So to start off, we'll look at the gene toxicity AOP. There are 2 pathways here with evidence associated, so I've looked at each one separately. So each piece of evidence was assessed and deemed acceptable. So we think the studies were of good enough quality that we trust the results that come from them. So on pathway A, there's 1 key event and it has 2 results associated. Both are negative. So it's easy to come to a conclusion as to its activity. For the chromosomal structural damage on pathway B, there are multiple data, and there's also a mix of results. So potentially we could take a very conservative view, and that if there's any positive data at all, that could give an overall outcome of positive for the key event. However, it might also be sensible to take results from in vivo into account over those from in vitro or in silico. So if we take that biological complexity into account and think about how it relates to what we want to get out of this, then we think this outcome again would be negative, as the in vivo result is negative, and we think this trumps the other results. So finally for the micronucleus formation, there's only 1 piece of evidence, so we take that result. As for the other 2 AOPs, there's only 1 piece of evidence for each, and this comes from in silico models and give positive results for each AOP. In this scenario, we want to assess human relevance, as this is important for an overall overall result. So there is information in the models, which we've linked here, which suggests that these are only rat-specific mechanisms. Therefore, the AOP relevance to human has been classed as unlikely in these cases. And then we saw these AOPs come together. So for the common key events in each pathway, there's evidence of organ weight increase from a subchronic study, which is associated with hyperplasia, as well as hypertrophy and peroxisome proliferation in the PPAR AOP. So if we want to bring all these results together, we could use the evidence to say clofibrate is positive, but a result is unlikely to be human relevant and therefore is unlikely to pose a risk in humans. So while there are positive outcomes for H2HR binding and PPAR binding, both these pathways is classed as not likely to be human relevant, and this is based on the information from the models we discussed earlier, which were actually direct nexus alerts. And additionally, if you look into other the evidence from a subchronic study, it suggests that the organ weight increase observed was likely because of hypertrophy, and this alone is unlikely to be indicative of progression to tumours. So these gross findings are consistent with rodent-specific mechanisms suggested from the in silico profiling. So we're quite happy with assessment. However, this shouldn't perhaps be the be-all and end of your research. You could go into more assays, can do more assays according to what you see along pathways. And you can show that this avenue research will need exploring further and to just add confidence to this hypothesis. This can also be then presented in a well-structured and transparent way so that it can be followed as to how a conclusion was reached. So that's where we've got to so far. So what does the future look like for us? So with many assays and predictions, you get a binary result. But there are usually measurements of potency captured when recording results. So if we want to move from a hazard scenario to a risk assessment scenario, we need to start thinking about how we can use these potency measures in combination with exposure to predict a risk. We could place these measures on the AOP, and these could help us to predict how these may propagate through a pathway and reflect in the adverse outcome. Additionally, the integration of ADME information can contextualize potency when extrapolated from in vitro to in vivo. Eventually, this might be used to work out how exposure to a certain chemical relates to the dose at which toxicity is observed, and thus how this translates to risk. So, in conclusion, I hope that has shown that AOPs can be used for organising evidence and help someone reach a decision as to the potential of a substance to cause unwanted effects. With this in mind, we have developed an AOP network for carcinogenicity, which repurposes information contained within direct nexus, public literature, and associated relevant evidence. This is captured in a new piece of software we call CAPTIS, and this is an AOP framework which can be used to organise and give context to data and visualise data and knowledge relationships with the aim of aiding decision-making in risk assessment. This framework has potential to be applied in various industries and settings, from discovery to safety assessment, and to answer a variety of questions with data and evidence organised in such a way to help make decisions in a transparent and robust manner. So with that, I'd like to thank the LASA Limited team. So all these people have been involved in various stages from putting together the knowledge, bringing it out of Derek and making prototypes, software, and bringing it all into our CAPTIS software. So with that, I'd like to thank you, and I'd also like to thank the organisers for inviting me to speak today. So thank you very much.
Thank you very much, Susanne. That was a great presentation. I really like seeing how you're applying not only one AOP, but multiple AOPs into these assessments. So we have quite a few questions in the Q&A. I'm going to try to switch back and forth between you both to make sure that we can get some questions for each of you. So I'm going to start with Martin. One of the questions on OncoLogic is, and I remember seeing in your slides a definition of the different terms like equivocal versus low to moderate. Are those available on the website so that people can understand how to distinguish between some of those terms?
Yes, I would strongly encourage people who are interested in those concern levels to check out the supporting information that's available on the OncoLogic EPA website. There's actually quite a bit of mechanistic information that also contributes to those concern levels. For instance, it is using an initiation, promotion, progression model of carcinogenesis, and chemical substances that are known to hit one or two or all three of those steps get different ratings depending on which they are hitting. So I would really encourage you to go and look at the website for OncoLogic.
Great, thank you. And so for Susanne, we have a question about the software and how or whether it considers the duration of MIE activation as a factor in leading to cancer, because AOPs number 37 and 41, the PPAR and AHR indicate duration as a factor.
Yeah, so we've not considered this so far, but as part of this, how we want to go from this hazard assessment to risk assessment, duration would be a big factor in going from that binary method to this continuous method. So it doesn't as yet, but that is part of our ambition.
Yeah, okay. Yeah, there was another question about quantitative information, and I think that's sort of along the same lines of what you mentioned as a next step.
Yeah.
Yeah. Okay. So for Martin, there's a couple of questions that kind of fall into the application of OncoLogic related to mixtures. So can you use OncoLogic for mixtures? And also, what about chemicals that are not in the classes? What happens to those types of chemicals?
So it will tell you if it's not part of a chemical class that OncoLogic currently supports. So in those cases, this software isn't going to generate a prediction.
Okay, that makes sense.
And for mixtures, it really can, it's because all of the predictions are structure-based, you will need structures in order to run the software. You can't generate it directly on a mixture. You would need to run it separately on each component.
Okay, that makes sense. Okay, Susanne. There's a question about ontology. Is there the models that you're developing and using, do they have a controlled vocabulary represented by ontology?
This is something we're also looking to. So we tried to standardize our terms in-house, but then we obviously want to relate them to standard ontologies used externally. So that is something we are working on at the moment as well. Yeah, I can't say much more than that at the moment, unfortunately. But yeah, that is definitely, particularly if you want to bring in data yourselves in a certain format, then we need to be able to link both our ontology to different ontologies to bring it all together and make it smooth without having to do a lot of fiddling, basically, to change terms, etc.
Yeah.
So yeah, it's something we want to do. Yeah.
It's a big question within the AOP wiki itself, and some efforts are going into that as well. So. Okay, a question for Martin. Are there examples of carcinogenicity assessments using OncoLogic for decision making, either standalone or as an integrated approach with other assays?
That's a great question. we are now posting redacted human health reports from our new chemicals program on regulations.gov. And so I would have to do a search to find some cases that have been posted on there where the human health report has the OncoLogic report in it. But the answer is we are using OncoLogic predictions in our new chemicals program on an ongoing basis. I guess I would add, so OncoLogic is, again, a refinement of the general structural alerts that we were using before, and we use it in the absence of data. So obviously, if we have data on a new chemical substance or a very close analog that indicates that something is likely to be a carcinogen, we will make our hazard ID taking that into account. But this is just really a way to incorporate all of this SAR knowledge in one easy to use tool that can be sort of run by our contractors or run by our assessors who are toxicologists rather than chemists, and get a result and a justification that they can use as part of the process. So it's one piece of the hazard ID.
And I imagine because it's available on the website, it would probably be a good tool for submitters to consider running themselves so that they kind of have a sense of, you know, what their submission might come up as.
Yes, we used to have a program called the Sustainable Futures Program, and we encouraged submitters to run OncoLogic on their new chemical substances and submit the report to us as part of their package. We haven't given the Sustainable Futures training in a while, but there are still submitters who have taken that training and still do submit OncoLogic results. So that is ongoing, and that's another way that we consider OncoLogic if it's provided as part of the package the submitter includes. What was I going to say? Oh, the Sustainable Futures Manual, which gives you specific instructions on how to run it on a new chemicals submission and sort of how we interpret it in the context of the new chemicals program, is available online. So that information is also available from EPA.
Okay. So for Susanne, well, there's a question about how the AOPs generated by LASA align with those being generated by OECD. But I think you're harmonized with OECD, right, in terms of using the AOPs that you're considering are in line with OECD's AOPs, right?
Yeah.
Yeah. Okay. Another question for Susanne: how could data on chemical toxicity against specific tissues like bone marrow and liver be incorporated into the AOP or into your software platform, or maybe are they?
Again, it's, yeah, so when we've been developing our AOPs, we've been putting context on these AOPs and the key event relationships as well. So where the results come from. So if it is bone marrow, be linking to a certain thing, then we can link it to a tissue of concern, and then these propagate through. So if it's bone marrow specific, for example, what you see in liver tumors, perhaps you can dismiss that result because it's not, you might not think it relevant to the in vivo effects you may be seeing or the in vivo effects you may be predicting. I think that, does that make sense?
Yeah.
Yeah.
I think so. Yeah.
Yeah. So yeah, we're thinking of context as we go along. I mean, with the AOPs developed so far, and with the models, it's been based on like data from Ames tests, which are done in bacteria. So it's like you had to be able to extrapolate that relevance going into the AOPs, or from rodent studies. So you've got to extrapolate that relevance. So if it's a rodent specific mechanism, we can contextualize that and then perhaps in future add some sort of confidence level, say, well, you might see it in rodents, but you probably might not see it in humans, that sort of thing.
Yeah, great.
Mm.
So I think, let's see, we have Martin, there's a couple questions that I can give you together. They're related to uploading or using an API connection to search a batch of chemicals or a table of chemicals rather than single chemicals. Is that possible?
Yes, I believe so. I am not as directly involved in all of the programming. However, I know that there is an API for OncoLogic because it needs that in order to communicate with the toolbox. So if someone's interested in integrating some sort of query with the API for OncoLogic, I can try and get you in touch with the software developer that might have more of the technical details. You know, feel free to send me an email and I'll get you directed in the right spot with that. As far as batch searching, it's not something that we're going to add to the standalone version of OncoLogic because our plan for the development of OncoLogic is to actually integrate it fully into the QSAR toolbox. And the QSAR toolbox has more batch searching functionalities. So rather than having it as a standalone software program that has a backend interface with the toolbox and maybe draws on its databases, it'll actually be something that it'll be one more profiler in the toolbox. I can't tell you how soon that's going to happen, but that's the reason why we're not pursuing more batch search functions at this point.
Okay, yeah, that's helpful to know. Thanks. So I think we'll take just one last question from for Susanne. We've got a few questions still, and I'm sorry we didn't get to all of them, but we can follow up. The speakers have graciously agreed to follow up by email so we can get to all of them. So just one last question for you, Susanne. Can you explain how non-linear carcinogens can be assessed using the tools that you have developed?
Can we elaborate on non-linear carcinogens? You're talking like non-gene toxic carcinogens.
Yeah, I think that would be an example.
Yeah. So we do have pathways for both, and at the moment it is very much a binary outcome. But so what I showed in my example, we did have that non-genotoxic pathway on the go. So I think I showed you an example of that as we're going along. Non-genotoxics are a lot harder, and I guess this again is when this whole the potency work would be really useful, because when it's non-linear, then you get unless it gets chemical smash into the system, then the mechanisms at play will write everything. So we'd have to think about how much exposure, how potent the chemicals are before the next effect would be observed. I mean, at the moment, we've just put the AOPs in place and we're starting to think about how that would happen. With genotoxicity, it generally is more linear, but not always linear. But yeah, with the non-linear ones, that the whole exposure, the mechanistic load, and chemicals and amount being put in would have to be taken into consideration. So yeah, we'd have to think a bit more about that and putting those sort of measures in place and using more models, I guess, to predict what those tipping points would be.
Yeah, I think you know, in terms of further AOP development that's necessary as well. So a lot of the AOPs right now are qualitative, as you said, and having a more quantitative understanding of the sort of response to response relationships of how one upstream key event, or under what circumstances, what amount of disruption for one key event has to happen in order to lead to the next key event. Some of that information, I think, is going to be necessary before we can have a more quantitative understanding.
Definitely,
Okay, well, thank you so much again to Susanne and to Martin. Thanks to Todd and Gino and Amy and our PETA Consortium hosts. It's been a great webinar. Thank you all for your attention, and I hope you have a great day. Thank you.